Hello and welcome to Nikolay's genetics lessons, and today's problem is given the template DNA sequence as follows, which sequence would be used by researchers as antisense molecule. And today's video is going to be very interesting because this is not just a video about finding antisense molecule, but application of this technology is very important in, for example, searching for functions of genes. I will explain later. And this technology also very important in creation of genetically modified plants or animals, in which this antisense technology would work like extra immune response mechanism. So I will explain in just a few minutes, and now let's find solution of this problem. Imagine that we have double-stranded DNA here, and here we see transcription bubble, and here is our messenger RNA. So this is mRNA, and this is double-stranded DNA. And of course 5' end is going to be here. Why I'm saying of course? Because messenger RNA is always made continuously, and as you remember, new strand of the DNA or RNA would grow continuously only from 5' end to 3' end. So all messenger RNA would start from 5' end and would grow in the 3' end direction. So that's how I know that this is 5' end. And if I know that this is 5' end, I can give you other ends of the DNA. So this is going to be template strand of the DNA. So we should have 5' end of the DNA here, and 3' end of the DNA here, 5' end of the DNA here, and 3' end of the DNA here. And in our problem, as you see, we don't have no 5' end, no 3' end, but usually all the sequence is given in the 5' end to 3' end direction. So those we do not see here no 5 no 3' end, but we have to assume that it have to be on the left side and 3' end have to be on the right side. So let's plot the sequence here on this template strand of the DNA. So this is going to be A, T, T, A, G, G, and C. So that means on the messenger RNA we are going to get following sequence: G, C, C, uracil, uracil, adenine, adenine, and uracil. Let's now imagine that this messenger RNA is dissociated from template strand of the DNA, and now we have this messenger RNA here, and 5' end here, 3' end here, and the sequence is G, C, C, U, U, A, and U. And now, in order to cancel this messenger RNA, we need another messenger RNA that is going to be complementary to this messenger RNA. So it have to go in the different direction. So have to have 5' end here and 3' end here. And the sequence have to be complementary, so uracil would base pair with adenine, uracil, uracil here, adenine, adenine, and guanine, guanine, and cytosine. So this is going to be sequence of the complementary messenger RNA that would make a double-stranded RNA, and mechanism of the cell would degrade such double-stranded RNA, because in the cell we do not have double-stranded RNA except of the cases when a cell would be infected with double-stranded RNA virus. So virus can be double-stranded RNA, but in our organism we do not have double-stranded RNA, and internal mechanism of the protection of the cell would recognize it instantly and would degrade such double-stranded RNA. So let's now find which the answer it is. So we have 5' end AUU. So here is answer A, AUU. So this is going to be once again 5' end and 3' end here, and we apply this 5' end to all these sequences. So let's check the first one. AUU, AUU, AA, AA, and GGC. So GGC, GGC. So right away we have found that the first answer is the correct answer. And by the way, even before we started to solve this problem, we could exclude variant D of the answer. Why? Because here we see thymines, and in messenger RNA, thymines would be substituted with uracil, and the same is here. So we can cross out right away these two answers. So if you would be short on time on your exam, it's better to exclude right away these two variants, and if you would run out of time, just would choose any of these answers, so you would have at least 50% chances to hit the correct answer. But this is not the end of my video yet. As I promised, here is the technology, how gene cancelling works. Imagine now that we have DNA sequence here. Here is going to be our gene that we want to cancel. Of course, this gene would produce messenger RNA, and we would have many such sequences in the cell, and each such messenger RNA would be used by ribosome to build a protein. But now imagine that we inserted the other sequence, DNA sequence, in reverse order. So now we have another messenger RNA that is going to be complementary to the first messenger RNA, and they would make double-stranded RNA, as you remember, which would be destroyed by the cell mechanism of protection, so we wouldn't see this product, this product, this product, and this product. But as you see, some of the messenger RNA still can be used in order to make a protein, and this is very important for search of the functions of the genes. As you know, the first variant of finding the function of the gene would be to make a deletion, single point deletion or insertion, and this would lead to a disruption of the sequence of the gene, and also within about 20 nucleotides we expect that this gene or this messenger RNA would hit a stop codon. So we would have a short version of the gene that would be non-functional. But the problem with this technology that not many genes can be cancelled completely, because organism wouldn't develop at all. So many genes are just that important that we just cannot delete them completely. So how we can find their function? And we can find their function using this antisense technology. We just greatly reduce the product that such a gene would produce, and product would be proteins, and now we would be able to see phenotypic changes, and we would be able to find the function of this gene. And the second variant how we can use this technology, imagine that this is plant cell, and here we have plant virus that would attach to the surface of the cell and would inject single-stranded RNA, and if we would insert antisense gene in the genome of this plant, this gene would produce messenger RNA that is going to be complementary to this RNA. And now mechanism of the cell would recognize it as a foreign material and would destroy it. Why organism wouldn't destroy it in the first place? Because this RNA would look just like any other messenger RNA that's present inside the cell in thousands and millions of copies. But now when antisense RNA would attach to it, the cell would recognize this as a foreign RNA that is normally not present in the cell, and cell protection mechanism would destroy and degrade such double-stranded RNA. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.